not. So I hope you've had a chance to look around and see our facilities. So not only do we have this breakout area here, but thanks to the Australian shareholders, we have refurbished custom-made labs and offices upstairs. And hopefully you've had the lab tours, but if any of you really wanted to see the lab, we have a break between 3 and 3:30. Amy, do you want to wait? Yeah, Amy is our lab manager. See her and she'll give you a bespoke, but very rapid VIP tour. Housekeeping loos are down the corridor as you come out to your right, just on your left, and also if you turn right, there's further down there. The programme for today is to have now a science session, but when I say science session, I hope it's going to be a user-friendly science session, because I'm aware that many people here are not scientists, and really the whole point of today is to actually share with you how Neuro-Bio is developing, why we're special, and I hope for you to feel that we are on the verge of a very exciting time. Every single person here, every single one of you, has contributed to Neuro-Bio. That's why you're here. Every single one, whether it's as a supplier, or as a member of staff, or as an investor, or as a collaborator. And you'll hear more from our collaborators as the afternoon unfolds. But I just want to now say thank you very much for all you've done so far, and perhaps all you'll be doing in the future. Okay, so I'm going to start off with a very quick, about 30-minute overview of where the science is. This will be followed by Sara Garcia-Rates chairing a panel discussion of our university collaborators, and this will be interspersed by some videos from people who sadly couldn't be here because they live in Melbourne or Barcelona, or they're not available at the moment, they're outside of the country. And then we'll have a tea break from 3 to 3:30, and then the second session will be the more wider aspects of the company, the corporate context, if you like, and I'll be wrapping up at the end. The reason we're having it on this day of all days is the stars were aligned in that two of our Australian shareholders, who've been very established and very helpful, Peter Yates and Martin Meyer, both independently but coincidentally happen to be in London. And moreover, and even more coincidentally, Todd Thompson from Kairos Ventures, who are VC funders, also happen to be in Europe. So it was obviously meant to be, it was obviously in the stars that today was the day when we should get everyone together. And I think it's a testament to Neuro-Bio that so many of you are here today. I hope we'll have a chance to talk more informally if you don't know me that well and want to have a chat. Not only during the coffee break, but when we go to St Hilda's, and I think most people in this room are signed up for the dinner at St Hilda's tonight. A bus will be leaving between 5 and 10 past 5. Amy May again is going to be the hostess on the bus. For those of you who don't have transport, can you see her? But I think we've got the names already. So our big fear is that someone's going to be left in the loos or something. Be stranded here. So let's try and avoid that contingency if we can. When we get to St Hilda's, there'll be an hour and a half for people to arrive, a reception accompanied by live music, thanks to Sarah's husband, a group called No Horses, for reasons that mystify us all. And then we'll have a dinner with further speeches, and then dancing for those that feel so inclined. So that's an outline of the day, and I think I won't waste any further time because it's very important that we do stay to time by introducing you to the science, and with apologies, to the demon scientists here, if I talk in rather non-specialist terms, and also to those of you who do know the story quite well. I thought it was nonetheless important everyone was on the same page for where we are. So, as you know, we are pioneering a new approach to Alzheimer's. And what's the story so far with Alzheimer's, which is a devastating condition characterized by confusion and memory loss? 44 million globally diagnosed, 9.8 in Europe, UK rising up to 100 to a million soon, USA 6 million already. And as you might expect with such a devastating condition, you can see the brain, by the time Alzheimer's is advanced, is in pretty poor condition. The dogma, and I stress the dogma, has been that this is attributable primarily to something called amyloid plaques, which are histochemical markers in the brain that frequently characterize post-mortem the Alzheimer's brain. And so you can't blame people. Quite obviously, you think, well, if this is a feature of the Alzheimer's brain, let's target that, and that would be perhaps a cure for Alzheimer's. And these are the amyloid plaques. People have tried this for a long time now, but the most recent was Aduhelm, was bought out by Biogen. You may have heard in the news in the summer. It was actually licensed by the FDA. It's aducanumab, which is an antibody, that's to say something that is going to intercept the amyloid and sort of neutralise it. So far so good. It's just that it's always been surrounded in any event by controversy. As you can see, there were resignations, and already people were slightly suspicious of its efficacy. And sadly, the last few months have been littered with lots of negative reports from that, and not least one where some scientists were discredited for fudging their data. So, on the one hand, this was bad news because it's always bad when science is shown up in such a light, and it also shows that when people adopt a dogma, how so many people subscribe to it without really challenging it too much. You may be familiar with a philosopher called Kuhn, Thomas Kuhn, who said that what happens in science is you have a dogma, this being a good example. You have a dogma, and everyone subscribes to the dogma and they build their careers on it and they put lots of money into it, but gradually anomalies creep in. And first of all, you try and sweep them under the carpet. They're one-off exceptions, but slowly more and more anomalies accumulate. So in the end, you can't ignore them. And that is when you have what he calls a paradigm shift. And I think we're in line now for a paradigm shift in Alzheimer's. Whether we are the people to come up with the alternative, of course, only future will tell, but I'm obviously biased, I think we are. So why has it been so long to develop a treatment? Well, this emphasis on brain amyloid, as you can see, one of the problems with it is you can see it as a feature of normal brains, and therefore we need to really step back and ask, well, if it is amyloid, well, why? How? What does it do? You can't just see it, it's not parachuted in, it must be there or has evolved there or be there for some process. And what we need to know is the mechanism. We need to know not the end product, the downstream consequence. What we want to know is why do brains embark on degeneration? Why does that happen? And because I've been initially at the universities, I'm sure most of you know, Oxford, where you give tutorials all the time and you ask questions, you have time to stare out the window and to join up the dots and to read the literature at a somewhat, I won't say leisurely pace, but you have time to do those things. And that's what I used to do until 2013, and that's what gave rise now to the novel theory that Neuro-Bio has been pioneering. So as you know, we were founded in 2013. It's obvious we're based here, you're here. We now have 17 patent families. I'm very proud of that. And Anton Hutter will be talking about those after the coffee break. This covers 6 independent assets, and I love this quote from an investor, which I think sums us up, which is, not only is our story highly interesting, but it has real novelty. It's a proprietary position, and it meets both the clinical and diagnostic utility with a seriously big unmet clinical need. So why are we so different? Well, our paradigm shifting approach is based, as I said, from time spent just in the library at Oxford and just reading and thinking and staring out the window. And what's evolved from that is, I think, a narrative, and I think we're the first people or the only people to offer a true explanation of how Alzheimer's happens that actually accounts for all the known clinical facts. And, for example, answers the question, how come, and this is one of the problems with developing a drug for Alzheimer's, how come it's underway for 10 or 20 years before you see the symptoms? Why? Why is that the case? And it strikes me pretty obviously, you should ask why. Everyone accepts it and knows this, but why? Why should that be the case? Well, here's the answer. It's because only certain brain cells, we believe, are the primarily vulnerable ones. And they have distinct profiles. Now, all too soon, and all too often, people have rushed to think that all brain cells are the same, that one size, rather like these ill-fitting suits on these gentlemen, one size fits all. And they mistakenly, and I think this is one of the problems with other, I won't call them even theories, other approaches to Alzheimer's, is it's not backed up by any theory, it's just, oh, it's there, it characterises the brain, let's target it. And the assumption is that all brain cells are the same, they're all generic. And this is the problem I would have with so-called approaches that look at inflammation, for example, which is a very generic response to damage. But they have different properties, and again, this seems to have been ignored until recently. And what we need to do is target these vulnerable cells. And then, if we knew what mechanism it was that was already operating proliferating and the vulnerable cells, we could start to understand why you don't see symptoms for 10 or 20 years, and then we could even intercept and deal with the problem before you have the cognitive onset. So this is our approach, is to look, joining up the dots, at the primarily vulnerable cells. And that would explain the inefficacy of most drugs to date, because the primarily vulnerable cells turn out very rarely to show amyloid. It follows, if you are basing your approach on amyloid, you are missing the party for 10 or 20 years. This is a bit of self-publicity, if you'd like to read it, from an interview. It's a transcript of an interview I did even before the discrediting of the scientists at the University of Minnesota, where no one could say Neuro-Bio has ever changed horses in midstream. We've always said the amyloid theory was not so much mistaken, but not appropriate or not sufficient in that it was downstream. So I hope this doesn't look too technical. Let me introduce you to the vulnerable cells in the brain. This is how you started life in the middle. This is a 4-week human brain. And you can see it divides into 2 levels or plates, the alar plate and the basal plate. And what's really exciting is that this basal plate gives rise to the cells shown roughly in the red circle there, which are kind of core cells in the brain. These are the cell groups which are linked with arousal, with sleep and wake. They're very diffuse. They're like fountains in the brain. I used to call them just that, the fountains. Other people have other names. One is isodendritic core. Doesn't matter what you call them, but they are distinct, and they are already at 4 weeks after conception, they are already different. And for me, the light bulb moment was a review that highlighted this. This review didn't actually acknowledge or know that these were the primarily vulnerable cells, not just in Alzheimer's, but motor neuron disease and Parkinson's. And it strikes me that surely that's nature giving you a clue. You have a primarily vulnerable group of cells, and they have a different embryological provenance. Shouldn't we explore what they do that's different from what other cells do, and that might give us a clue to why they degenerate. And what's very exciting is they selectively have retained, even into adulthood, the ability to grow again, a developmental mechanism. That sounds very exciting when I say that it explains the mechanism of so-called excitotoxicity. That means to say that in normal development, something that promotes growth, that is excitatory, in the context of a mature brain, is a bit like Jekyll and Hyde. The same mechanism now will kill cells. I won't necessarily go into the details unless you want me to in the questions, but it's something called excitotoxicity. It's where there's a spectrum of Jekyll and Hyde. You go from excitatory to toxic. And these cells have such a mechanism because they have retained it into the mature brain. And they also have a high degree of redundancy, as might befit very primitive cells. So, for example, in Parkinson's disease, you often have a co-pathology of Parkinson's and Alzheimer's. You have to have 80% of the key cells there have to die before you have the motor impairment. So that tells you these are very primitive cells, they are very all tangled up, they all work as a group, and it's only when a lot of them are damaged that eventually you'll see a problem. Hence, the 10 to 20 year time lag. So we can explain that, and we can explain the co-pathology of Alzheimer's and Parkinson's. So already then, it makes these cells very interesting. And what is particularly interesting about them is they all contain, and you'll hear this name over and over again until 11 o'clock tonight, it's T14. They all contain a chemical messenger called T14. So you might ask, what is T14? It's a peptide, that means that it's a fragment of a protein, and it comprises 14 little building blocks of amino acids, hence the name 14. What does it do? Well, it promotes cell growth, and this is a collaboration we've done, and it's on one of the posters, I think, with Nadine Kabani at George Mason University. You can see here how the cell has kind of little fingers of so-called lamellipodia, and as you can see here, compared to the controls, which are untreated cells or a different, just an ordinary old peptide, compared to that, our T14 has had a dramatic effect on cell growth. And you can see that here in this rather lovely picture, shown very obligingly with the oblong blocks. When does it happen? Well, in perinatal brain development, you can see P stands for postnatal. 7-day-old brains have high degrees of T14, and that declines into being 60 days of age, and that correlates perfectly with the activity, as shown here, with something called optical imaging, which again we have in the lab, some of you may have seen that, where you can look at the activity of whole groups of brain cells, where red is very active and then declining is the perimeter. So we have evidence that indeed LT14 is a feature of the developing brain and that it indeed promotes growth. And to that end, we have now a good target. And Anton Hutter from Venner Shipley will talk about this much more. If you look at the number of patents for amyloid, you can see it's 2,500. Anti-tau, which is another marker, 894. Anti-T14, 6. And we own more. So if we've cornered the market, if you like, in this approach. Well, I hope we have. Anton will elaborate more. So the key molecule that drives growth can be, as I said, at Jekyll and Hyde. It depends on the amount. So you can see here low levels of T14. You can, I hope, see these little whiskery outgrowths indicated by the red arrow. We'll call that the Jekyll situation. But it becomes Hyde as you increase the dose, or for the amount you apply it. You can see here in green a short application of 1 hour has encouraged growth of the cells, so the neurite outgrowth. When applied for 24 hours, the opposite effect, it's now toxic. But for what's the most revealing for us was not our own work, but again reading a paper published in the late 90s, and they showed that there was a difference between whether it was a growth or it was lethal with age. Okay, and it seems that the same amount of agent that promotes cell growth and development will be toxic if that system is reactivated in maturity. And that's what we think happens. That's the Jekyll and Hyde situation. Okay, so what we'd like to suggest is neurodegeneration, and we have a story, we have a narrative. It's an aberrant form of development. It's when that is triggered, and T14 could be the active driver of that. So what we're suggesting is, if you look at the Alzheimer's profile, there's a chemical called G1, which is an indirect measure of the T14. Again, I won't go into details of what G1 is. Happy to do afterwards, but just believe me, trust me. And you can see here, the G1 in this peak here is high in the embryo in development. Indeed, it's also high in Alzheimer's. So that shows you, again, that Alzheimer's is a recapitulation of development, and this G1, which is an indirect measure of T14, is high in both cases, so you can see them in between the two. In the youngest brains, you can see here the T14 is high, and here you can see it recapitulates, and these are age-matched post-mortem Alzheimer's brains. You can see in the controls in black, it's half what it is in the Alzheimer's post-mortem brain, and this is in the area of the brain, the so-called midbrain, which are these vulnerable core cells. So, shown here. So if we look at these vulnerable core cells, one of them being the substantia nigra, which degenerates both in Alzheimer's and in Parkinson's. If you look at the rodent substantia nigra, and let me introduce you to Zoltan Molnar, who's sitting on the front row here. This is do you want to just stand up, Zoltan, so they see you? Just so there's Zoltan. He's going to be part of the panel discussion, and his assistant, August, and we've been collaborating with them looking at the substantia nigra, which is called black because it has melanin in human cells, which is a black pigment, insulin. And this is what Zoltan and August have been showing, that the T14 overlaps very closely with dopamine, well, almost every cell, and that when it's activated, it can be released from these cells. So this is a quiet cell, then reactivates it. You can see the green showing more T14. So we know that the T14 is in the dopamine-containing cells of the substantia nigra, and that it can be released as a so-called signaling molecule. Working then with clinical samples, and this with Gregory Cole at UCLA, who's one of our collaborators, thanks for an introduction from Kairos. What he's shown is that in the early stages of Alzheimer's, so-called Braak stage 2, the T14 is partially filling the cells, there's lots of them. By the time you get to the later stage, so-called Braak stage 5, you can see there's far fewer cells and they're much more intensely stained. So it seems that the T14 is characterizing the death of these cells, and as you can see, if you look at the T14 relative to the number of cells, that it is actually increased as Alzheimer's is advanced. So this is what's called proof of concept, that indeed the T14 is operating in this region. So let's just recap. These are the normal cells. The white cells shown here are the hub containing the primarily vulnerable cells for both Alzheimer's and Parkinson's. And remember, they have retained, since their embryo provenance, they have retained the ability to grow again, unlike other cells in the brain. The Jekyll situation. That said, for the moment, because it's a normal situation, the mechanism is dormant, the T14 is there, but it's not really doing anything. It's a healthy brain. Imagine now if that is damaged. Now, if other parts of the brain are damaged, other cells will compensate. If these cells are damaged, because they have retained the ability to grow again, they will mobilize the T14 mechanism, because after all, that promotes growth, so this will help them grow. But that's the very worst thing that they could do, because this is in the context now of the mature brain, and this is Jekyll. So now the cells will die. And then more cells will die, because it's like a snowball effect, it's a feed-forward mechanism. So there'll be more and more damage, and that will spread throughout this area, and as often occurs, then have both Alzheimer's and Parkinson's occurring at the same time. And it will spread throughout that region, but still nothing will have happened, because remember, these cells have a high degree of redundancy. So all of us now could be sitting here in this situation. And it's only when, finally, the damage spreads to other more sophisticated areas of the brain that you will see amyloid and you will see memory impairment, and that is when someone will go to the doctor. And that is when drugs will start to be prescribed. So if we're right, you can see, is it no wonder there's been no successful treatment for Alzheimer's? If action only occurs after that red arrow. And really, what we need to look at is this T14 molecule, it's this Jekyll molecule. So that's our basic theory. Why has it been so long? Well, as I say, it takes 10 to 20 years. MCI stands for marked cognitive impairment. And clearly, what we should first do is think of a way of monitoring the T14 before that red arrow, before we get beyond BRAC stage 2, before we get to the symptoms. And we're not alone in acknowledging the need for this. This is someone you might recognise. The current process for diagnosing Alzheimer's is a huge hurdle standing in the way of breakthrough. We need a cheap, non-invasive way to diagnose patients early before their symptoms get too bad. Okay, so that's what we're trying to do. So this is the Neuro-Bio vision. Okay. Imagine being able to go to the doctor, like you might for a cholesterol test, and you could have a test for T14. And this might be blood or skin or even a lateral flow. We'll hear more about that later. But if that was for your age, and having been characterised, indicative that the neurodegeneration is already underway, and in an ideal world, you could then have a blocker of the T14, if indeed we're right, and that means if that was given at the pre-symptomatic stage, then you'd never get the symptoms. And this would be a cheap, relatively painless, NHS nice compliant way of treating Alzheimer's, and I would argue it's an effective cure because you'd never have the symptoms. Even if you already had the diagnosis, it would stabilise soul loss. And even if we didn't have the drug, having early diagnosis, you'll see in your packs, there's a leaflet from Alzheimer's Research UK on the merits of having early diagnosis. So let's think first about the biomarker. Where is Neuro-Bio with the biomarker? Well, the obvious place to look is blood. And we have indirect evidence. You may recall I mentioned G1 as an indirect marker of T14, and indeed in Alzheimer's blood it is very high, so that suggests T14 is there. We've also worked with a company based in Portugal called ILOF, standing for Intelligent Lab on Fibre, and they have a method that doesn't use antibodies, but it's machine learning and optical imaging, and they've already detected at the picomolar range T14 in the blood. That needs a lot of refinement. Or we can use antibodies, and this was some very early work done in-house, where you can see the T14 in the blood was increasing as Alzheimer's progressed. So what we need to do, and what we did then, was to look at Alzheimer's and how it compared with normal aging. And in this early study, with relatively few samples, but it still showed a trend, you can see that with normal aging, and remember this, the T14 seems to decline with normal aging in the blood. However, with Alzheimer's, it was a reverse trend, it seemed to increase. I'll come back to T14 being indicative of aging and declining with normal aging, and that being reversed with Alzheimer's. So that's one, that's where we are with the blood. Another idea has been linked from something you may have read in the papers recently about a lady who could actually smell her husband had Parkinson's disease, bizarrely enough. And that was related to the sebum, which is the oil in the skin, shown here. And indeed, that does suggest that perhaps there might, bizarrely, be a link between skin and Alzheimer's. Now bear in mind the skin is one of the few parts of your body that is in constant development, that's in constant cell renewal. So it could just be that the same mechanism, they both come from the ectoderm, both brain tissue and skin, the epidermis comes from the same place. It could well be the cells there behave in a similar way to the brain, and therefore we could have a biomarker of T14 in the skin, and that could reflect Alzheimer's. You can see here someone else's work showing the old markers amyloid and tau in disarray in Alzheimer's, and here I'm not going to read these in detail, just look at the bold bits. This is the literature, not our work at all. You're very welcome to this talk afterwards, by the way. All of which points to a link between Alzheimer's and skin. Yeah. So Jason Harcutt from Unilever will be talking more this afternoon about our work. Where's Jason? Is that here? Just stand up, Jason. Just say. This is Jason Harcutt from Unilever, with whom we've been collaborating on TV, and he will. I don't want to. He'll be talking about the possibility of T14 as a skin biopsies and how it might affect aging. Another possibility is saliva, and this is probably illegal, but among friends who said, just for fun, what we decided to do was spit into, 6 of us just spat into some tubes, just for fun, and Georgia's here, Georgia Price, who pioneered this. Where's Georgia? Where's she gone? Oh, she's at the back there, hiding, because she ended it illegal. So just 6 of us did for fun. We thought, can we measure the sort of T14 in saliva? So this is utterly uncontrolled, it's utterly not regulated, utterly unquantifiable, but it's just something you do on a Monday morning. And sure enough, there you are, the red arrow is indicating the T14. We then did a control to eliminate the possibility that it was recognising something else by pre-incubating the antibody with the antigen, the T14, and you can see there's nothing there. So this is a very, very high indication that T14 might be measurable in saliva. All of this points then to the possibility of a lateral flow test for T14, and Chris Hand from Abingdon is in the front row. I won't ask him to stand up, I'm sorry, but he's here, and he will be talking this afternoon about possible collaboration developing a lateral flow test pre-symptomatic for T14. But let me just set the scene. We know that this idea has occurred to others for measuring amyloid. It's a shame that amyloid is such a late-stage marker. But nonetheless it shows it's possible. And the reason it's possible is came with some surprise that a large contribution into nasal secretions comes from cerebrospinal fluid. That was disparagingly called the urine of the brain. That breaks the brain and spinal cord, and therefore about 50% of it finds its way into nasal. 50% of nasal secretions are made up of CSF, so it would be a very intimate and good place to look for what might be happening in the brain. And indeed, when we've measured our T14, you can see here that CSF in Alzheimer's postmortem postmortem CSF, there's clearly an increase in Alzheimer's CSF. And more recently published, and this was the most exciting, that if you look at the progressive stages of Alzheimer's characterized by 1 and 2, which are the pre-symptomatic stages, 3 and 4, which are mild cognitive impairment, and then the later stage, full-blown dementia. No one else, as far as I know, and unless anyone tells me I'm wrong, no one else has identified a marker that has changed at perhaps stage 1 or 2, and as you see we have in the brain. So all we have to do now, is translate that profile that is in the post-mortem brain into, let's say, nasal secretions or saliva, and then develop that as a lateral flow. That's all we have to do. And that could be seriously a game changer for early diagnosis for Alzheimer's. And that is going to be what Chris Hans is going to talk about this afternoon. Okay, so much for where we are. My ultimate dream would be if you could develop a lateral flow, which is socialised already, it's low cost, you don't need medical supervision, it's painless relatively, it's frequent, we've all had them, it's fast. It's so fast that perhaps eventually, if it was highly quantitative, the amount that was measured of your T14 in your lateral flow could inform by readout how much drug you then had to take. This is the drug, MVP. But it would be amazing because that would be personalising medicine, and it would be a kind of Goldilocks approach whereby you weren't taking too much or too little of the drug every time you took it. So that would be another huge benefit. So MVP14, this is our drug. We're now getting onto the therapy. How does it work? Okay, so this is T14, a little scheme of it. And it binds to something called a receptor, which is a protein in the membrane of brain cells. and it binds to a site, so-called allosteric site, preventing toxic excess calcium going in. So it's a little bit like, sorry, no, this enhances it. This is the T14. It's a bit like someone with their hands up doing evil things. It's a headless man doing something evil. What the drug does, it's like tying the hands behind their back, inactivating it by binding it into a circle. So for those scientists here, and especially the chemists, please forgive this complete ravaging of chemistry. But I hope you get the idea that we have this molecule that will enhance in a toxic way calcium entry, but by so designing it to be inactive, to have its hand tied behind it by being in a cycle, it's now inert. And what it will do, it will nonetheless bind to where the normal T14 should be, but it will displace the evil T14. So therefore, it won't do any harm. It's a bit like someone with evil intent trying to sit in the chair where someone else is sitting in the chair, but their hands are tied behind their back. So the person in the chair can't do anything, but nor can the person with evil intent. So that's how it works. And so NBP14, our drug, our prototype drug, which found for the company, is nothing more nor nothing less than the evil T14 itself, but just chemically inactivated. And here it is in action, displacing the toxic T14. So these are in brain slices, obligingly symmetrical about the central axis, where when you intubate one side with the drug, you can see the T14 has indeed been displaced, as it has in the human brain post-mortem. This is Gregory Cole's work. As the dose increases, so it's displaced it. And when you give it so-called in vivo, that is to say to a living mouse in this case, and it was administered intravenously, just one dose, you can see over a certain time period that 4 hours maximum is displacing the naturally occurring agent. So we are quite confident of the action of T14 and how it is working. So does it actually block the toxic effects? Yes, in culture, you can see here, amyloid also has an effect on the cells, enhancing calcium, but the MVP has actually blocked it. The cell viability, the number of cells that are alive, are reduced with the toxic peptide, the toxic T14, 30, and with amyloid, the MVP's reversed it. And if you compare it with galantamine, which until aducanumab came along was the licensed drug, our drug has, unlike galantamine, has no side effects. It's 100 times more effective at blocking A-beta, and it's 50 times more sensitive at binding to the target. So we're quite confident that our drug is good. So we want to process it now to clinical trials, and I'm delighted that Georgios is here from Bachem. Where's Georgios? Yeah, Georgios at the back is here from Bachem. I hope I'm not misquoting you, Georgios. It now has clinical level purity, 97%, and the yield was 4 times higher than was expected. So apparently the synthesis for a peptide has been surprisingly promising. Evotec, who are an international company that work with living animals, which we can't do, then took on looking at the toxicology with intranasal delivery. They found no adverse side effects, any of the doses tested, and the so-called pharmacokinetics showed, that's to say, the way it's distributed in the body, how long it hangs around, how it's distributed, was detected in blood for over 2 hours, which gives it the properties of a well-absorbed drug. We're now in conversation with various companies for so-called formulation, that is, devising it into a nasal delivery system. As yet, because it's a resource issue, we're still just talking to these companies, but the boxes you have to tick to go to clinical trials are API production, which is what Bachem are doing, safety and toxicology, which is what Evotec are doing, and formulation, which is what we're discussing now. So we're trying to do everything we need in parallel to bring this drug to phase 1 clinical trials. Does it work? Well, obviously, we think that this is very promising. When Evotec gave it twice a week to mice, you'll see the effects. Now, there were 3 groups of mice. One group were normal mice, so-called wild type, they're pale blue. The dark blue group were the transgenic animals, so-called. These were animals genetically tweaked to have Alzheimer's. And then there was the MVP-treated animals. Now, all are performing above chance, recognizing novel objects at the beginning of the experiment. But 14 weeks later, it's a very different picture. You can see that the ordinary mice are learning well above 50%, they're doing well. But the Alzheimer model that have been treated with just saline are now lamentable. They're just at 50% recognition, they're not memorizing anything. Whereas the same type of animal treated with our drug twice a week for 14 weeks are performing similar way to the normal animals. So this was for us a huge breakthrough and very exciting. And when we looked in the brains, you can see the amyloid, which is a downstream marker shown here as gold blobs, was markedly reduced in the drug-treated group. So this has given us the confidence to go forward. How did it compare with aducanumab? Well, you can see in terms of efficacy, we actually know how it might work. It can actually halt death rather than just slowing things down. We had an outside agency do a rough cost estimate, and they came in at £6,000 a year as opposed to 40. The mechanism of action is actually targeting the real mechanism that causes the problem as opposed to blocking a downstream component. We know it gets into the brain because of the effects we're seeing, and it could be given intranasally at home, either by a carer or by the patient themselves, as opposed to having to spend a whole day in hospital with all the stress and expense that would involve. So, why is there still no effective treatment? Well, again, the issue is with animal models. Until you know what the basic mechanism is, then you can't reproduce that in a rodent, and that halts research hugely into developing a new drug. This has been in the literature, lamenting the problem. And so these animals, the ones that we had to use ourselves, are transgenic rodents. They're very expensive. They necessitate a lot of time waiting for the behaviour to kick in. They don't replicate authentically the clinical profile. And that has been understandable because no one else until we have found the basic mechanism. However, now, working with the University of Nottingham, and Tobias Bass will be part of the panel discussion from Nottingham, and again with Evotech, in a preliminary study where we gave the evil chemical into ordinary mice, rats, you can see that there was a decline in neuron number compared to the controls. Amyloid beta was increased and tau was increased. So that is very promising. It does suggest, yes, there's frank cell loss, just like in early Alzheimer's. Increased amyloid and tau. Amyloid plaques, no, because there's no plaques in early stage. Memory loss, not for the first 6 weeks. And we are suggesting this might replicate the model of early pre-symptomatic Alzheimer's. So our narrative is that we have a basic biological process driving cell growth and renewal that continues in normal healthy skin. If this goes wrong, it can lead to neurodegeneration or metastases or skin disorders, that's hyperproliferative skin disorders such as psoriasis or eczema. So cancer very quickly, we have a link to cancer as you're here. You can see here with strongly metastatic cell lines, shown in red here, are the two groups that were treated with MVP in culture, and you can see that has slowed down the rate of metastases to those similar to non-metastatic cell lines. And we're now working with Robin Anderson and Rick Revis, who you'll hear from in a video, who are based in Melbourne at the Lever Newton-John Centre, and they've been working with very aggressive metastatic cancer, breast cancer cell lines shown in the yellow arrow. And you can see here that in a dose-dependent way, looking at primary tumour growth, as the increase of MVP, as it's increased in dose, so the primary tumour has been reduced. But very exciting, more recently, you can see the metastases in the lung. When they looked at the post-mortem lungs, the cancer cells that had spread, you can see there were none in 8 out of 9 of the mice that were treated with MVP, none, that just compared to the control. So we're, as you might imagine, very excited. The other group that will work with cancer, you'll hear from a Genopat, and again, you can see here with colorectal cancer, very promising effect with a low dose of MVP in reducing primary tumour growth. So our narrative then is this basic biological process, and you'll hear, as I said, about the skin from Jason Harkup. And so for the first time, we think that we can offer to you an empirically validated narrative for how Alzheimer's occurs. And why should we be successful when other approaches are failed? Why should we be so different? Well, it's because we actually, I think, have a narrative. And I want to quote from one of my heroines, Rita Levi-Montalcini, who you may have heard of, an Italian lady. You can see her dates, which coincided with the rise of fascism in Italy. She was Jewish. She was practicing medicine. Because she was Jewish, she was banned from practicing medicine. And so what she did was set up a lab in her own home, and the work she did there then led to the Nobel Prize. And she see how beautiful she was as well. And she said, above all, don't fear difficult moments, the best comes from them. And I like to think that's what we're doing. I'd like to end with two self-promoting quotes from Richard Mose, who was on our science advisory panel, a novel, innovative and potentially very fruitful approach to treatment of Alzheimer's. And from Paul Herling, who sadly can't be with us, but you'll see a video in a moment, has the potential of a breakthrough for treatment for Alzheimer's disease. So I'd now like to introduce you to to one of them, Paul Howling, and to Gary Manchi. Neither could be here today, but there's some videos, and we're going to start with Gary Manchi, who, as you can see here, has been advising us on taking the drug forward to clinical trials. He had 30 years' experience at GSK, and I'll introduce you to him now, I hope.
Hello everyone, I'm Dr. Gary Manchi, and I represent Neuro-Bio on the development of NBP14 for Alzheimer's disease. And also, I represent Neuro-Bio in other exciting therapeutic areas. I would like to share with you some of the great progress that has been made to date and where Neuro-Bio plans to go in the future. Alzheimer's disease represents a huge unmet clinical need, with over 55 million people worldwide living with dementia in 2020. This number will almost double every 20 years, reaching 78 million in 2030 and 139 million in 2050. At present, there is no real disease-modifying treatment for Alzheimer's, and even the current gold standards are at best poorly effective. NBP14 is a new, extremely exciting cyclic peptide which prevents the endogenous ligand T14 from interacting allosterically on the alpha-7 nicotinic acetylcholine receptor. Inhibition of T14 by NBP14 results in a lower influx of calcium into cells, increasing their survival, particularly in those brain regions responsible for Alzheimer's disease. Amazingly, and as equally exciting, is the finding that NBP14, given intranasally, was shown to be efficacious in an in vivo mouse model of Alzheimer's disease. The availability of disease-modifying treatment for Alzheimer's would ultimately be a game changer and is really the therapeutic prize. For Big Pharma, finding a game-changing drug has proved very costly and time-consuming, with the proof of concept extremely difficult to validate. However, Neuro-Bio have addressed these issues, and as a consequence are proceeding on two fronts. Firstly, Neuro-Bio are developing a disease-modifying drug, NBP14. And secondly, Neuro-Bio are developing a novel, non-invasive and relatively inexpensive biomarker for Alzheimer's disease. In this way, Neuro-Bio is addressing both the disease biology and its early diagnosis, which together will address the current challenges for Alzheimer's. So where is the Alzheimer drug NBP14 currently? Well, the new developable acetate salt of NBP14 has successfully been synthesized and has been scaled up to provide sufficient material for initial toxicity testing, which incidentally the drug has already successfully passed. We found that after intranasal doses, no toxicity was seen at the highest concentration possible, which is equivalent to 30 milligrams per kilogram. However, this was 3 times higher than the previous efficacious dose. Interestingly, no toxicity was observed even at 30 milligrams per kilogram. But this is encouraging nonetheless, and also a pharmacokinetic profile was observed in blood after a 30 milligram per kilogram intranasal dose, which gave a drug half-life in blood of approximately 1 hour. So where to next for NBP14? NBP14 is currently being investigated on 3 fronts, effectively. Firstly, NBP14 is already in preclinical development. We intend to obtain a repeatable, efficacious daily dose, to progress with more toxicity testing, and to explore further our future drug requirements for longer-term toxicity studies. Secondly, NBP14 is being used to progress in other indications like cancer. And lastly, further studies are ongoing to support initial exciting data seen in 2 types of animal cancer models. NBP14 has shown efficacy in both breast and colorectal animal models. And lastly but not least, NBP14 is being used to explore backup molecules. other molecules are currently being investigated in order to underpin the position of NDP-14 in multiple therapeutic indications. In summary, these are exciting times for Neuro-Bio. We have embarked on the development of NDP-14, a new exciting molecule with a novel mode of action for Alzheimer's disease. And it has the potential to stop this disease even before symptoms emerge. This, together with the availability of a non-invasive biomarker, represents a true breakthrough in Neuro-Bio. Lastly but not least, the involvement of T14 and its antagonism by NDP-14-like molecules has already been implicated in other disease states, like cancer and skin ageing, which opens up many opportunities in the future. In conclusion, I believe Neuro-Bio stands on the edge of several major breakthroughs in terms of biology, diagnosis and molecular diversity, which together puts Neuro-Bio in good stead for future investment. And thank you for listening.
Okay, so that's Gary Manchi, and the next person I'd like to introduce you, who sadly couldn't be here, is one of our non-executive directors, Paul Herrling, who's based in Switzerland. Nonetheless, he's been very helpful and enthusiastic and supportive of us, and he suggested, because he didn't want to, he said it's like doing a selfie, which he abhorred, he didn't want to make a video, but what we settled on was showing a clip from a press conference that we gave in April, where he, Paul Herrling, actually promoted the idea that although we're very novel and disruptive, that that will have to be the way we go. So this is Paul Herrling.
Fascinating to me here is that, having followed Susan's work for many years, because I was in the field, and it was a completely different approach, but consistent with the